Welcome to the deep dive. We take all the crucial knowledge from a whole stack of sources and bring it straight to you.
That's the idea.
Today, we're diving deep into genome editing, where things stand right now. We've got this really comprehensive report, covers everything, from the early tech right up to the cutting edge, clinical trials, the works.
Yeah, it's a packed field.
So our mission, really, is to give you a clear picture of this transformative field, the breakthroughs, the challenges that are still there, and just the sheer potential of being able to, precisely change the code of life.
And it's not just medicine, right? It goes way beyond.
Exactly. Inherited diseases, cancer, sure, but also agriculture, biotech. It touches so much. We know you want the essentials, those aha moments, without getting totally bogged down. That's what we're aiming for.
Happy to help unpack it. It's amazing how fast this field moves. I mean, from theory to actual therapies changing lives. It's pretty remarkable.
Okay, so the report kicks off looking back at the early days, wanting precise genetic changes. It mentions restriction enzymes. What was the issue there? Why did we need something more?
Right. Restriction enzymes were foundational molecular scissors, basically. But they only cut at specific sequences that naturally occur in DNA.
So you couldn't pick the spot.
Exactly. You were limited to where those natural cut here signs were. The big goal was finding tools you could program, tools you could tell, go cut right here, anywhere in the genome. That need for programmability, that's what drove the next steps.
And that led to zinc finger nucleases, ZFNs. How do they actually work?
Okay, so ZFNs are engineered proteins. Think of them in two parts. First, you've got the DNA binding part made of these things called zinc fingers. You can string these fingers together to grab onto a specific DNA sequence.
Like little hands gripping the DNA?
Sort of, yeah. And the second part is attached to those fingers, a nucleus, an enzyme called foci, that does the actual cutting. Now, the clever part is foci only works when two of them come together, when they form a dimer. So you design two different ZFNs, one for each DNA strand, right next to each other.
Ah, so they have to team up.
Exactly. When they both bind, their foci parts meet up and make that double-strand break. And needing two of them, that actually helps with specificity, making it less likely to cut the wrong place.
So back then, what made ZFNs seem so promising? What were the advantages?
Well, a couple of things. They were relatively small. Their size was a plus. That mattered for gene therapy using viruses, which can only carry so much cargo.
Right, the delivery vehicle.
Yeah. And maybe more importantly, they were the first tools that really showed potential in human clinical trials. There were early studies for HIV trying to block the virus getting into cells, and for some single gene disorders. Sangamo Therapeutics, that company, was a real pioneer there.
But the report says there were some pretty big hurdles with ZFNs, too. What were the main difficulties?
Designing those zinc finger arrays to be super specific, that was tricky. It wasn't always straightforward to get them to bind only where you wanted. Off-target cuts were a real concern.
Cutting in the wrong place.
Right. They developed ways to try and minimize that, like tweaking the foci part so it would only pair up with a specific partner, but it was still a challenge. Plus, getting enough of the ZFN into cells, potential toxicity if you use too much, and the patient's immune system may be reacting to this foreign protein. Those were all big issues, too.
Okay, but despite those limits, the report suggests ZFNs aren't totally gone. They're still relevant, even seeing some good clinical results lately.
That's right. CRISPR definitely took over in many labs because it's just easier to use, but ZFNs have found their niche, especially for certain therapies, often in vivo therapies, meaning directly inside the body.
Because they're smaller for delivery.
Exactly. That size advantage still counts with certain viral vectors, and Sangamo's progress is really impressive. They've pushed forward with HIV, hemophilia B, MPS I and II, and their Fabry disease work with isaraldigene civaparvovec or ST920, that's looking really good.
What are they seeing there?
Long-term data. Years after just one treatment, patients still have sustained enzyme levels. Many can stop their old enzyme replacement therapy, and they're seeing kidney function improve. Plus, the safety looks favorable. They're even talking to the FDA about potentially getting accelerated approval.
Wow, okay.
So yeah, ZFNs were absolutely vital as proof of concept. They showed this could work, and they're still making real progress in specific areas. Shows what focused effort can do.
All right, next up, chronologically. Talons. Transcription activator-like effector nucleases.
Yeah.
How are they different from ZFNs?
Talons were the next big step. Like ZFNs, they use that foci nuclease to cut the DNA. The difference is how they find their target sequence. Instead of zinc fingers, they use DNA binding parts from tau effectors proteins from certain plant bacteria.
Okay, different binding module.
Right. And the big breakthrough with talons was the simplicity of their binding code. It's much more straightforward. Each repeating unit in the tail domain recognizes one specific DNA-based pair, like a direct one-to-one code.
So easier to design, more predictable.
Yeah, much more predictable than designing ZFN arrays. It was like having a simpler instruction manual. And because they usually bind to longer DNA sequences than ZFNs, they also had the potential for higher specificity, less chance of off-target binding. Plus, they worked well in lots of different organisms.
But they must have had downsides, too. The report mentions limitations.
Definitely. A big one was size. Because of those repeating units, talon proteins could get really large. That made delivery tricky, especially with viruses like AAVs that have tight size limits.
The delivery challenge again.
Yep. And those repetitive sequences could sometimes be unstable when you were building the talons in the lab. And even though the design principle was simpler, actually making and testing functional talons could still be more work and more expensive than, say, making a CRISPR guide RNA.
So where did talons make their mark? And what's their status now?
They were used successfully for lots of things, knocking out genes, precise edits using a template. We saw them used in agriculture, like making disease-resistant rice or hornless cattle, and in basic research, figuring out what genes do.
But then CRISPR came along.
Exactly. CRISPR's ease of use, efficiency, and especially its ability to hit multiple targets at once, multiplexing meant it largely took over for many applications. But talons are still valuable. If you need extremely high specificity, or if you're working in an organism where CRISPR has issues, they're still a great tool. They really helped broaden the field and set a high bar for specificity.
Which brings us, like you said, to the CRISPR-Cas revolution. This sounds like where things really took off.
Oh, absolutely. It was a total game changer. Realizing that this natural immune system in bacteria could be repurposed for editing genomes so precisely and, relatively speaking, so easily, that shifted everything.
Bacterial immune system. How does that work?
So bacteria and archaea use CRISPR-Cas to fight off viruses. They have these CRISPR regions in their DNA with repeating sequences and these unique spacer sequences copied from viruses they've fought off before.
Like a most wanted list?
Kind of.
Okay.
When that virus shows up again, the cell makes RNA copies of those spacers, cRNAs. These cRNAs team up with Cas proteins, forming a complex that finds the matching viral DNA and destroys it. It's like microbial adaptive immunity.
And the key was hijacking that system, telling it what DNA we wanted it to find and cut.
Precisely. The genius, especially with the class 2 systems we use for editing, like Cas9, Cas12, is that they use a single protein guided by an RNA molecule. For Cas9, the workhorse, you engineer this guide RNA and sgRNA. Part of it matches the 20-letter DNA sequence you want to target.
So you just change that 20-letter sequence to aim it anywhere.
Pretty much. It's like changing the coordinates in a GPS. You just swap out that guide sequence, and you can send Cas9 almost anywhere in the genome you want. It made targeting incredibly straightforward.
The report gets into the nitty-gritty of Cas9 and Cas12. Can we break down how they each operate? Let's start with Cas9.
Okay, SpCas9, the one from Streptococcus pyogenes, is the most famous. It's basically two parts. The Cas9 protein itself, which is the scissor molecule with 2 cutting domains, and the single guide RNA, the sgRNA.
The GPS.
Right. It has the targeting sequence, that 20 base bit matching the DNA, and a scaffold part that holds on to the Cas9 protein. But there's one more critical piece, the PAM sequence.
Yeah.
Protospacer adjacent motif. It's a very short DNA sequence right next to the target site. For SpCas9, it's typically NG any base, followed by 2 guanines. Cas9 needs to see that PAM sequence to know it's in the right place to cut. Think of it like a specific docking site.
So no PAM, no cut, even if the guide RNA matches.
Exactly. Once the Cas9 sgRNA finds the target DNA and verifies the PAM is there, then it snips both DNA strands, creating a clean, blunt cut.
And how is Cas12 different, like Cas12a?
Cas12 systems have some key distinctions. They also use a single protein guided by RNA, but Cas12a, for instance, can actually process its own guide RNAs from a longer transcript, which is neat. Their PAMs are different too. Cas12a usually looks for T-rich PAMs, like TTTN, located upstream of the target.
Upstream, okay.
And when Cas12 cuts, it makes staggered ends, leaving little 5' overhangs, not blunt ends like Cas9. That difference in the cut can sometimes be useful for encouraging certain types of DNA repair or insertion. Plus, some Cas12 variants are naturally smaller than SpCas9, which helps with that delivery issue we keep mentioning.
Okay, so once Cas9 or Cas12 makes that double-strand break, what happens then? How does the cell fix it?
The cell has its own repair crews that jump into action. There are 2 main pathways we care about for editing, NHEJ and HDR.
NHEJ.
Non-homologous end joining, that's the cell's fast emergency repair system. It basically just glues the broken ends back together. But it's often messy, error-prone. It frequently introduces small insertions or deletions. We call them indels.
And indels can mess up a gene.
Right. If an indel happens in the coding part of a gene, it can scramble the message and knock out the gene's function. That's useful if you want to disable a gene.
Okay, and the other pathway, HDR.
Homology-directed repair. This one is much more precise. It uses a template, usually the sister chromosome, to accurately repair the break. For gene editing, we can trick it by supplying our own DNA template, one that contains the change we want to make.
So you provide the correct sequence.
Exactly. If the cell uses our template during HDR, we can get precise corrections or insert new sequences. The catch is HDR is generally way less efficient than NHEJ, especially in cells that aren't actively dividing, which is most cells in our bodies.
That inefficiency seems like a big hurdle for corrections. But overall, it sounds like CRISPR had huge advantages that made it take off. What were the key ones?
Oh, huge. The biggest was simplicity. Just changing that short RNA sequence to retarget, that was revolutionary compared to re-engineering whole proteins like ZFNs or talons.
Much easier.
Way easier. And CRISPR is generally very efficient at cutting. Plus, you could easily target multiple genes at once, multiplexing, which opened up totally new ways to study complex biology. It was also cheaper, more cost-effective, which democratized the technology.
And it wasn't just for cutting DNA, right?
Yeah, right. You could use a dead Cas9, one that binds but doesn't cut, called dCas9, fuse other proteins to it, and you can use it to turn genes on or off CRISPR or CRISPR, or edit epigenetic marks, or even light up specific DNA spots in living cells. That versatility is incredible.
Okay, but like any powerful tool, CRISPR isn't perfect. The report spends a lot of time on the limitations and challenges. What are the big ones?
Yeah, especially for therapies, we have to be really careful. Off-target effects are a major concern. That guide RNA sequence might be similar enough to other spots in the genome that Cas9 cuts there too, accidentally.
Which could be dangerous.
Potentially, yes. So there's been a ton of work on minimizing this, better guide RNA design tools, engineering high-fidelity Cas enzymes that are pickier.
Pickier Cas9s.
Yeah, variants like SpCas9, HF1, HyperCas9, things like that. Also using anti-CRISPR proteins as an off switch, or delivering the Cas9 and guide RNA as a pre-made complex, an RNP, which doesn't stick around as long in the cell, reducing the time window for off-target cuts.
Another thing mentioned was the PAM requirement again. How does that limit things?
Well, needing that specific PAM sequence right next door means you can't just target any 20-base sequence in the entire genome. If the PAM isn't there, standard SpCas9 won't work.
So it restricts your options.
It does. That's why finding new Cas enzymes from different bacteria with different PAM preferences like SACAS9 or Cas12a was so important. And scientists have also engineered Cas9 variants like SPG or SPRY that are less picky about the PAM they need. But sometimes tweaking the PAM recognition can affect how well it cuts on target or even its overall specificity, so there can be trade-offs.
And then there's delivery. Getting the CRISPR system into the right cells in vivo, still a major bottleneck.
Huge bottleneck. Viral vectors like AAVs are common, but AAVs can't carry huge payloads, which is tough for bigger Cas proteins or complex systems. Lentiviruses carry more but integrate into the genome, raising safety questions. Plus, the immune system can react to viruses.
What about non-viral methods?
Lipid nanoparticles, LNPs, are really promising, especially for delivering mRNA that tells the cell how to make the Cas9 and guide RNA. They've shown great success targeting the liver, but getting LNPs to other tissues efficiently is still a big challenge. Delivering the RNP directly is another option. It's transient, maybe safe for long-term, but getting those big protein RNA complexes into cells in vivo is tough.
Immunogenicity, too. Our body's reacting to these bacterial proteins.
Absolutely. Our immune system sees Cas9 as foreign. This can neutralize the editor before it works or cause inflammation. So people are looking for less immunogenic Cas versions from different bacteria, using transient delivery like RNPs, hiding the components in stealth nanoparticles, or sometimes using immunosuppressants.
And back to that HDR problem, getting precise corrections is still tough because NHEJ is more common.
Exactly. Especially for therapies needing correction, not just knockout. HDR efficiency is often low, particularly in non-dividing cells. Lots of effort here. Trying to temporarily block NHEJ, syncing cells to phases where HDR is more active, optimizing the donor DNA template design using SODNs, single-stranded templates, seems to help, or even fusing Cas9 to HDR-promoting proteins.
And the last risk mentioned is the DNA break itself triggering problems,
Hmm.
like DNA damage responses or bigger genomic changes.
Yes, making that double-strand break isn't totally benign. It can trigger cell defense mechanisms. Sometimes cells pause growth, sometimes they die, apoptosis. There's also a concern, though maybe rare, about selecting for cells that have lost crucial safety genes like p53. And while unlikely with good design, there's a theoretical risk of unintended large deletions, vector insertions, or chromosomes getting rearranged near the cut site. These are serious potential issues needing careful study and monitoring.
So despite all that, CRISPR clearly changed the game in research, and now the focus is shifting towards making it safer and more precise for actual therapies.
Exactly. The first wave was about the power and ease for lab work. Now it's all about refinement for the clinic. Developing those high-fidelity Cas versions, expanding PAM compatibility, and moving towards next-gen systems like base and prime editing, alongside better delivery. That shift from protein-guided ZFN stalins to RNA-guided CRISPR was huge, and we're still building on it.
Okay, let's dive into those advanced systems.
Yeah.
The report really emphasizes base editing as a way to get precision without the double-strand breaks. How does that work?
Base editing is really clever. Instead of making a full cut across both DNA strands, base editors directly change one DNA letter into another at a specific spot. They do this using a fusion protein.
A fusion. Two proteins stuck together.
Right. You take a Cas9 that's been modified so it only cuts one DNA strand, we call that a nickase, or even a dead Cas9 that doesn't cut at all, and you fuse that to a deaminase enzyme. That's the enzyme that chemically changes the DNA base. The Cas9 part acts like the guide, bringing the deaminase to the right DNA address. When it binds, it opens up a little window on the DNA, and the deaminase can then modify a specific base within that window.
And you mentioned different types for different base changes.
Yeah, two main classes. Cytosine base editors, CBEs, convert a CG pair to a TA pair. They use a cytosine deaminase. Often they include another bit like UGI to help the edit stick.
Okay, C to T.
Right. Then there are adenine base editors, ABEs. They convert an AET pair to a GSC pair using an engineered adenine deaminase. So between CBEs and ABEs, you can make all the transition type mutations. And using a nickase Cas9 helps that single nick encourages the cell's repair systems to use the edited strand as the template, making the change permanent.
So what's the big win for base editing compared to standard CRISPR?
The main thing is avoiding those double-strand breaks. That means much lower risk of indels, those messy insertions deletions, less risk of larger rearrangements, less triggering of those p53 damage responses.
Sounds much safer.
Potentially, yes. You also tend to get higher product purity. More of the edited cells have the exact change you wanted, compared to relying on HDR after a DSB. And critically, they work well in non-dividing cells, which is a huge advantage for treating many tissues.
Are there downsides, limitations?
There are. One is potential bystander edits. The deaminase has that small activity window. It might edit other Cs or As nearby, not just the one you intended.
Collateral damage.
A bit, yeah. Also, they still use a Cas protein for targeting, so you're still limited by the PAM requirement for that Cas. And current base editors mainly do transitions. Other changes, transversions, are harder. Some early deaminases also edited RNA off-target, though newer ones are better. And being fusion proteins, they're quite large, which can be a delivery challenge.
But despite those, it sounds like base editing is really moving forward clinically. The report mentions specific companies and trials.
Absolutely. There's constant improvement, making editors more efficient, more specific, work with more PAMs. We've seen things like AB8, B4max, AVmax souped up versions. And companies like Verve Therapeutics and Beam Therapeutics are pushing hard.
Her is the cholesterol one.
Right. Using in vivo base editing via LNPs to turn down the PCSK9 gene. Their VRV-E102 candidate showed really promising initial results in humans lowering LDL cholesterol with good tolerability. They're planning phase 2.
Yeah, Beam.
Beam has a lot going on. Beam 101 is an ex vivo approach for sickle cell and beta thalassemia aiming to boost fetal hemoglobin. Beam 201 is an allogeneic CAR-T for leukemia, using multiplex base editing, editing multiple genes at once. And Beam 302 is really exciting an in vivo treatment for alpha-1 antitrypsin deficiency. Early data showed it actually corrected the disease-causing mutation in patients' liver cells. First clinical proof of in vivo precise mutation correction.
Wow, that's huge.
It really is. Base editing is a massive step towards safer, precise correction of those single-letter genetic typos that cause so many diseases.
Okay, another really cool one the report covers is prime editing, the search and replace system. How does that manage even more complex edits?
Prime editing is incredibly sophisticated. The goal is even more versatility, making all 12 possible base changes, plus small, precise insertions or deletions, again without a double-strand break, and, importantly, without needing a separate donor DNA template.
How does it do that?
It uses a different fusion protein, a Cas9 nickase fused to a reverse transcriptase enzyme.
Reverse transcriptase, like from retroviruses.
Exactly. An enzyme that makes DNA using an RNA template, and the key is the special guide RNA called a prime editing guide RNA or pegRNA.
pegRNA. What's special about it?
It does double duty. Part of it guides the Cas9 nickase to the target site, just like a normal sgRNA, but it also contains the template for the edit you want to make, plus a primer binding site, PBS, to kickstart the reverse transcriptase.
So the guide RNA itself carries the blueprint for the new sequence.
Precisely. Here's how it works, roughly. The Cas9 nickase cuts one strand. The loose DNA end then binds to the PBS on the pegRNA. The reverse transcriptase uses the template on the pegRNA to write the edited sequence directly onto that DNA end.
Wow, writing the correction right there.
Yeah. This creates a little flap of edited DNA. The cell's repair machinery then comes in, removes the original unedited flap, and incorporates the newly written edited sequence. Sometimes they use a second guide RNA to nick the other strand strategies like PE3 to encourage the cell to adopt the edit.
What are the big advantages of prime editing?
Versatility is the main one. Being able to do all base changes plus small indels potentially fixes a much wider range of mutations than base editing alone. And again, avoiding double-strand breaks should mean fewer unwanted indels. Plus, not needing that separate donor template simplifies the delivery package.
But it sounds complex. What are the challenges or limitations here?
It is complex. Efficiency can be a major issue. It's often lower and more variable than base editing or standard CRISPR, depending on the target and the edit. Those PE3 strategies to boost efficiency can sometimes increase off-target edits or cause larger deletions.
And delivery. That fusion protein sounds big.
It is big Cas9 plus a reverse transcriptase. So delivery, especially with viruses, is challenging. Designing and making the pegRNAs is also more complex than standard guides. And we're still fully figuring out all the DNA repair pathways involved, which makes outcomes sometimes less predictable. There's also ongoing work to check for any weird byproducts or off-target effects from the nicking or the reverse transcriptase activity.
Still, the report mentions lots of ongoing work to improve prime editing.
Oh yeah, the field is moving fast. Developing better prime editor versions, PMAX, PE2, PE6, PE7 with improved enzymes, optimizing pegRNA design, building tools to predict success. We're even seeing prime editors based on different Cas proteins like Cas12a, systems for bigger insertions like TJPE or PASSAGE, and even ways to do multiplex prime editing. Delivery is also getting better optimized dual AAVs, better LNPs, EVLPs.
So earlier stage than base editing clinically, but potentially very powerful.
Exactly. Prime Medicine is the main company pushing it towards the clinic. The versatility is incredible, but hitting the efficiency and delivery benchmarks of base editors is still the main goal right now.
It's amazing how the toolkit just keeps expanding. The report also flags some other novel tools and emerging tech. What else is on the horizon?
It's constantly evolving. There's huge interest in finding or engineering new Cas enzymes. Like the Cas13 family, they target RNA, not DNA. That opens doors for knocking down RNA. Editing RNA bases by fusing Cas13 to enzymes like ADAR or ApoBec or even diagnostics. Casrax is a nice compact Cas13.
And smaller DNA editors, too, for delivery.
Definitely. Things like SACAS9, CJCas9, and the really tiny ones like Cas12, also called Cas14 or Casmini or Ksa, Casx, making them small enough to fit easily into AAVs is a huge priority for therapy.
And what about these CRISPR-associated transposons, casts? They sound different.
They are really cool. These are natural bacterial systems that use CRISPR guidance to insert large pieces of DNA directly into the genome without needing HDR. Think precise integration of whole genes. Systems like MECAST or Integrate hold massive potential for delivering big therapeutic payloads accurately.
The report also mentioned more on RNA editing itself, beyond Cas13.
Yes, besides using Cas13 fusions, there's a really neat approach using our own cellular machinery. Specifically, ADAR enzymes, which naturally edit RNA in our cells.
How you direct our own enzymes.
You design special guide RNAs, sometimes called add-RNAs or antisense oligos, that bind to the target mRNA but have a mismatch where you want the edit. This creates a little bubble that attracts the cell's own ADAR enzymes to come and make the specific change, like A to I editing.
Clever. Any examples?
Yeah, R&E is developing this for alpha-1 antitrypsin deficiency. They use chemically tagged oligos to target the liver and recruit ADARs to fix the SERPINA1 mRNA. It's transient, reversible, potentially dose-adjustable. Preclinical looks good. Trials expected maybe 2025.
And epigenetic editors.
Yeah.
Changing gene activity without changing the DNA code.
Also super powerful. You use a dead Cas protein, DUCAS9, fused to an enzyme that adds or removes epigenetic marks, things like DNA methylation or histone modifications. These marks act like dimmer switches for genes, so you can precisely turn genes up or down without altering the sequence. Huge for research and potential for therapies like reactivating silenced tumor suppressors in cancer or guiding cell development.
And underlying all this, constantly improving delivery systems.
Absolutely critical. Better AAVs, engineered capsids for better targeting, less immunogenicity, sneaking past antibodies, dual AAVs using intine splicing to deliver bigger editors in pieces, better LNPs, targeted LNPs like Editas is working on for stem cells, virus-like particles, VLPC, EVLPs for RNP delivery, potentially less immunogenic, plus all sorts of other nanomaterials. Delivery is key to making all these amazing editors actually work in vivo.
It really paints a picture of incredible progress on all fronts, the editing tools themselves, and how to get them where they need to go. Okay, let's shift gears to the really exciting part. Therapies in clinical translation. How is this actually being used to treat diseases?
Right, this is where the rubber meets the road. And the impact is starting to be felt, especially in treating genetic disorders, developing new cancer therapies, and even tackling infectious diseases.
Let's start with those monogenic diseases caused by a single faulty gene. Seems like a prime target.
Definitely. Because you know the exact genetic problem you need to fix. We're seeing amazing progress, especially with ex vivo editing, taking cells out, editing them, putting them back. Sickle cell disease and beta thalassemia are the big examples here.
That's where Casgevy comes in, the first approved CRISPR therapy.
Exactly. Casgevy, exobgene autotemcel. It's approved now in the UK, US, EU, other places. It edits patients' own hematopoietic stem cells ex vivo to boost fetal hemoglobin production. Huge milestone. Over 90 patients treated, significant revenue already. It's real.
Are there others in that space?
Editas Medicine had a program, Renacell, but shifted focus now to in vivo editing for those conditions using targeted LNPs. And Beam Therapeutics has Beam 101, their ex vivo base editing approach, trying to achieve the same goal-boosting fetal hemoglobin via HPFH mutations.
What about in vivo treatments for other single gene disorders? The report lists quite a few making headway.
Yeah, in vivo is where the potential really expands. Intellia Therapeutics is a leader here. NTLA 2001 for ATR amyloidosis uses CRISPR-Cas9 via LNPs to knock down the TTR gene in the liver. Showed strong TTR reduction in phase 1, now in pivotal phase 3 trials. Got RMAT designation, looks very promising.
And they have another one.
NTLA 2002 for hereditary angioedema, HAE. Same idea, in vivo CRISPR via LMT to the liver, this time knocking out the KLKB1 gene to reduce kallikrein and prevent attacks. Phase 2 results were great, significant attack reduction. They've picked a dose for phase 3 and are aiming for a BLA submission in late 2026. Could be a functional cure.
Amazing. What about high cholesterol? You mentioned Verve.
Right, Verve Therapeutics. VRVE101, their first ABE candidate targeting PCSK9 in the liver, showed LDL reduction but hit some safety issues, so they pivoted to VRVE102. Same base editor, different LNP delivery system. Initial phase 1b data from April 2025 looked really good, significant LDL lowering, well tolerated. Phase 2 planned for later this year.
And alpha-1 antitrypsin deficiency. Beam's in vivo correction.
Yeah, Beam 302. That early phase 12 data showing actual in vivo correction of the PIZ mutation with ABEs, leading to increased functional AAT protein, was a landmark result. Intellia also has an in vivo CRISPR insertion approach, NTLA3-OBR1, in earlier stages, and Arena has that RNA editing approach coming too.
So multiple shots on goal there. Any others standing out?
Sango's ST920 for Fabry disease, that in vivo ZFN therapy using AAB delivery. The updated long-term data is compelling. Sustained enzyme activity off standard therapy, improved kidney function, good safety, potential accelerated approval path there too. And while Editas deprioritized their LCA10 program, ETA101, the initial work showed in vivo editing in the eye was feasible. DMD is still tough due to delivery challenges.
The report contrasts ex vivo versus in vivo. Can you quickly sum up the pros and cons and where things are headed?
Just vivo, take cells out, edit, put back. Pro, more control, safety checks before infusion. Con, Limited cell types, complex, patient-specific, needs conditioning. Casgevy is the prime example.
Okay. And in vivo.
Deliver editor directly into the body. Pro. Targets more tissues, simpler concept, scalable. Con. Delivery is hard, potential toxicity, immunogenicity, harder to control dose. NTLA 2001-2002 Verve's therapies, ST920 are examples.
So where's the trend?
Early successes are largely ex vivo. It proved the concept works. But the long-term push is definitely towards in vivo for broader impact. The liver is the low-hanging fruit for LNP delivery now. Next-gen delivery targeted LNPs for blood stem cells, better AAVs, is key to unlocking more in vivo targets.
Makes sense. Let's switch to cancer. How is genome editing changing cancer therapy?
It's having a huge impact, especially on cell-based immunotherapies like CAR-T, using CRISPR to engineer T cells to be better cancer killers, making them more effective or persistent by knocking out break genes like PDCD1, creating off-the-shelf allogeneic CAR-Ts from healthy donors by knocking out genes like TRAC or B2M to prevent rejection or GVHD, even inserting the CAR gene precisely into spots like the TRAC locus for better control.
Multiplex editing, too.
Yeah, hitting multiple targets. Beam 201 involves 4 edits. CRISPR Therapeutics has candidates like CTX112, CTX131 with edits for enhanced potency. And it's not just T cells. People are engineering NK cells too.
Beyond CAR-T, directly targeting cancer genes.
That's being explored too. Targeting ontogenes like CRAS or MYC, fixing tumor suppressors like TP53, using CRISPR activation, CRISPR, to turn up protective genes, plus using CRISPR screens to find new immunotherapy targets.
Mostly ex vivo for cancer right now.
Yes, most clinical trials involve engineering immune cells outside the body. Getting editors into tumors or immune cells in vivo effectively is the next big challenge, but a major goal. CRISPR is definitely turbocharging the ex vivo cell therapy field right now.
What about infectious diseases? How can editing help there?
Several ways. Directly attacking the virus's genetic material, Cas9 for DNA viruses like HIV or HSV, Cas13 for RNA viruses, or modifying our own cells to resist infection, like knocking out CCR5 for HIV resistance, which ZFN's tried and now CRISPR is doing.
The bacteria.
Using CRISPR to target essential genes or antibiotic resistance genes in bacteria is a really interesting idea, maybe delivered via phages viruses that infect bacteria. Plus, editing can help develop better vaccines.
How far along is this clinically?
Generally less advanced than for genetic diseases or cancer. Big challenges are delivery to all infected cells, especially hidden reservoirs like latent HIV, and the fact that pathogens mutate quickly, potentially escaping the CRISPR guide.
Okay, let's look at the overall clinical trial landscape. What did the report's analysis find?
Up to late 2022, they counted about 76 trials. Cancer was the biggest chunk, then viral infections, 19, mostly HIV CCR5, then monogenic diseases, 26, mainly blood disorders, shows CRISPR becoming dominant. Most are early phase 1 or 12 focus on safety and initial efficacy signals.
And the outcomes generally.
Increasingly positive. We've discussed the key results from Casgevy, NTLA 2001-2002, Verve, Beam, Sangamo's ST920, showing real therapeutic benefit is possible. But safety is paramount, constantly monitoring for off-targets, immune reactions, delivery issues, long-term effects of DNA breaks.
And the big success story, the approved therapy.
Casgevy, exigmoboglobin autotemcel. First approved CRISPR therapy for sickle cell and beta thalassemia, a true landmark. Lyfgenia from Bluebird Bio is also approved for sickle cell, but it's gene addition, using a virus, not genome editing like Casgevy.
So, summing up the clinical picture.
It's maturing fast. From ex vivo for rare diseases validating the tech, now pushing hard into in vivo for broader conditions, especially targeting the liver initially. CRISPR-Cas9 is clearly the main engine driving this therapeutic revolution right now.
This has been incredibly insightful, a real deep dive into a field moving at lightning speed.
Yeah.
As we wrap up, what are the absolute key takeaways you want listeners to hold on to?
I think number one is just the sheer transformative potential here. Across biology, medicine, it's huge. Second, seeing that clear arc of progress from the early tools like ZFNs and Talons through the CRISPR revolution, and now into these really sophisticated base and prime editors offering amazing precision.
And the clinical progress.
That's takeaway 3. It's not just theoretical anymore. We're seeing real therapeutic applications, real progress against tough genetic diseases, new angles on cancer, even potential for infectious diseases. Casgevy's approval is proof positive.
But challenges remain.
Absolutely. Delivery, off-targets, immunogenicity, those are still major hurdles being actively worked on. But the pace of innovation to tackle them is just relentless. That's maybe the final point, the sheer ingenuity and momentum in this field.
So, as we finish, a thought for you, our listener. Having this kind of precise control over the code of life, well, it brings up big questions, doesn't it? Ethical, societal implications. As you think about all this, what future developments seem most exciting or maybe most concerning to you? We hope this deep dive got you thinking and encourages you to keep following this vital area. Thanks for joining us on the Deep Dive.